Most Taoists somehow have an origin in microtubule work. There were cell biologists working on microtubules. Tau turned out to be a microtubule-associated protein. I mentioned that Tau is a microtubule-associated protein, so a quick word about microtubules. Microtubules occur in neuron. We have here a neuron with a long extension. That's the axon. That poses a transport problem for the neuron because things at the synapse have to be fed from the cell body. The transport system of the axon is, of course, the microtubule system. Microtubules can be thought of as tracks, and which have motor proteins running along them in the forward direction kinesin and the reverse direction dynein. And like a railroad track, it has to be stabilized by ties, and the ties for the microtubule system is the tau protein and other microtubule-associated protein. They're shown as black hooks here. So the hand-waving argument about the function of tau is that it stabilizes microtubules. It does actually a lot more, but that's the most basic function that was discovered early on by Mark Kirschner and colleagues. If the tau does not bind properly to the microtubule, for example, because it gets phosphorylated by certain kinases, then two things happen. The tau can come off the microtubule, and when it comes off the microtubule, it is then free to associate with itself for reasons we don't really understand, but anyway, in the course of that, it can form oligomers and paired helical filaments or tangles. And the other thing that happens is that the microtubules can fall apart, and therefore the tracks for transport disappear. So you have an obstacle in the brain that can inhibit axonal transport, and the tracks themselves are inhibited. In the year, I think, 85 or 86, it was discovered that the main component of the paired helical filaments in Alzheimer's disease was the tau protein. Pathological functions of tau. As you all know, tau is, is one of the deposits in Alzheimer's disease. It occurs in neurofibrillary tangles within neurons, in contrast to the amyloid plaques that are outside neurons. And if you isolate it, you get these filaments. They're called paired helical filaments. Here is a picture of the domain structure of tau. And the thing that I want to point out is this box here is the so-called repeat domain. 3 or 4 repeats. The green ones are alternatively spliced segments of tau. Anyway, the repeat domain is the one that interacts with the microtubules in a physiological sense, but it is also responsible for the formation of the paired helical filaments, and therefore the physiological function and the pathological function are encoded into the same part of the molecule. So it's like a switch, either pathological or physiological switch. A fantasy model of tau sitting in an unfolded way on top of the microtubule. And this unfolded structure shows you that it is an open, loose cushion sitting basically on the surface of a microtubule. Because of this highly soluble, open, hydrophilic nature of tau, it does not aggregate. And one of the points that I'd like to make is that tau by itself, and I'll show you some evidence in a minute, tau by itself does not aggregate. In spite of the fact that we see aggregates of tau in various brain diseases in a very stereotypical fashion, tau is so highly soluble, it has no reason to aggregate unless we assume that there is some other factor that helps the aggregation. And macromolecules may play an important event in helping tau to get on its way to aggregation. So tau by itself does not do any harm as far as aggregation is concerned, but in the contact with other macromolecules, it may do so. If you want to interfere with this toxic pathway of aggregation, then you can do two things. You can either knowing what the elements are that cause the aggregation, namely these hexapeptide motifs, you can either change those hexapeptide motifs so that they cannot aggregate, and one way of doing this, at least in vitro or in animal models, is to put in proline mutations because the residue proline is not compatible with beta structure. Now let me talk about the regular motives for controlling aggregation. I mentioned before the elements, the hexapeptide motifs that are responsible for the aggregation. These are elements at the beginning of repeat R2 and repeat R3. At the beginning of repeat 2 and beginning of repeat 3, you have these hexapeptide motifs, VQINK or VQIVYK, and if you mess around, they have an enhanced potential for beta structure. And if you change these peptides, then of course you affect aggregation. And Martin von Bergen, who discovered this in the lab, he coined the term pro-aggregant mutations and anti-aggregant mutation. The pro-aggregant corresponds to the mutation delta K280, which is observed in tauopathies, which enhances the beta propensity of this element. And the anti-aggregant simply, you stick a proline in here, you stick a proline in there, and you get no more aggregation. And the reason why this turned out to be important in our experience is that transgenic mice or transgenic worms or transgenic anything, if they have pro-aggregant mutations, they get sick. And if you just do the anti-aggregant, they stay healthy. So that is a very simple way of manipulating the state of disease. If tau cannot aggregate, and in fact this protein does not aggregate in these different animal models, then you have no effect of tau. You can overexpress it. There may be slight effects of overexpression, but the animals don't get sick. A few words about transgenic animals. This is mostly the work of Eva's group, so I'll just go quickly and leave out some slides that I don't understand. So here is, again, the tau structure with the hexapeptide motifs. This is where the delta K280 mutation is, and most of the models we made consist of placing pro-aggregant tau into a mouse or an anti-aggregant tau, and then comparing the two. And this is done in a inducible way, controlled by doxycycline, basically, so we can switch the expression of tau on, and then we can observe the aggregation of tau and the development of pathology, and we can also switch it off, which is a nice part of this application. So you switch the tau off, the tau disappears, and you can ask the question, does the disease go away? It does, actually. So now, first of all, showing you a pro-aggregant mouse, 3 months old, has a lot of tau deposits in the brain. An anti-aggregant mouse, even though it's 22 months old and very aged, shows absolutely no aggregation, which shows you the power of replacing a single proline in an aggregation-prone filament. You get rid of aggregation. So it's perfect cure on the on the level of molecular biology, at least. We have the same thing on the level of C. elegans. C. elegans moves nicely around, but when it expresses a tau in the neurons, and which gets aggregated, then it gets paralyzed. And we use the C. elegans as a cheap way of monitoring drug effects. I can't go on here. So I won't go into this, but just show you when they're paralyzed. This is where the mice have the C. elegans, the worms have aggregated tau in their neurons. But when you treat them with aggregation inhibitors, then they move quite nicely around. So this is an easy experiment to do, because the worms take up the drugs much better than, of course, the mice, because there's no blood-brain barrier. So you can check whether an aggregation inhibitor actually works. You can check this relatively quickly, and this works well for the delta K280 mutation. But I'm just